For the rest of the time available, we'll talk at a more basic level about collecting things like expression data and also information both at a RNA level but also at a protein level. And then I'll also talk about methylation, so what it is and how we measure it. But firstly, I'll just start with information about the regulatory aspects of the genome. And so a lot of that information is in the Encyclopedia of DNA Elements, which is otherwise known as ENCODE. So we know most of the genome is made out of these regulatory regions, and 90% of the GWAS loci for these complex traits and diseases are situated in these regions. Understanding how they're annotated and tested to infer function can really help understand the association, the mechanism and the cause. And an important note to make is that genetic information of an organism can be differentially expressed over time and in different tissues, and this can be influenced by genes, the environment, and of course their interactions. So if we just focus on DNA, the expression of RNA is controlled by a number of factors. So we've mentioned quite a few of these: chromosome accessibility, transcription factors, histone modification, DNA methylation, promoters. And enhancers, silencers, and also post-transcriptional modifications. And so ENCODE is a really good place to start to sort through these. The initial pilot phase of ENCODE started with 40 genomic regions, so 20 were quite well annotated and 20 were randomly chosen. Then they used a whole lot of different methods to actually define the unknown regions and say how good are they picking up what we already know. And then they've subsequently expanded this to include the entire genome. And recently they've published over 30 companion papers in Nature, Genome Research and Genome Biology, and they've created a what they've termed a threads website. And so if you visit this site here, there's all these different aspects that you can click on. It gives you links to the papers that are relevant to that particular aspect of the regulatory genome. This is a summary here that they also have of the different methods that they're using. So these are all complementary to try and understand different aspects of this genome regulation. These are kind of put together in and assembled into these databases so that you can interrogate them. So I'll go through some of the methods. So one I thought was quite interesting was thinking about DNA rather, often we see these Manhattan plots and it's in a linear form. But in actual fact, when we look at the DNA is not actually sitting in a straight line. We know it's folded, and these chromosomes actually sit in territories. So this particular method that they've used, they started at 3C, 4C, 5C. Now they're using chia-PET and HiSeq. So this stands for Chromosome Conformation Capture Carbon Copy. And so basically the way they do this is they take the nuclei, extract it out of the cell of interest. This nuclei, some of it is actually looped together, and so they're able to ligate the DNA, and then they tag it with biotin. This ligates, then they can fragment and sequence, and then that gives them a map about what the nuclei actually, how it was folded. And so from the contact maps that they've actually made using this technique, a lot of the contacts are within chromosomes. So, which is quite interesting. And then this figure here just shows a part on chromosome 7 that they've gradually zoomed in. I'm not exactly sure how they determine the actual the folds of that DNA, but they can actually map the regions that are linking together. And often this means that rather than genes talking to each other or next to each other, you know, from a linear perspective, they're hundreds to even several megabases away in the genome. And there's evidence to say that these peaks are conserved across both human and mouse cells. And occasionally gene activation is accompanied by an emergence of a cell type specific network of these peaks.
Actually, the question about the closest gene, this is actually why sometimes the caudal gene is not necessarily the closest to the G1.
Yep. And so chromatin profiling is obviously very important for kind of understanding the way that DNA is folded, and this is specific to certain cell types. And so the way that they work out how the DNA is folded is they use a combination of techniques. So this includes ChIP-seq, DNA-seq, ATAC-seq, MNA-seq, and FAIR-seq. ChIP-seq actually reveals the binding sites of specific transcriptional factors. So here you're actually using an antibody to enrich for a particular area to look for a peak. DNAs and ATAC-seq, from here you can see they actually reveal the regions of open chromatin. So these are the histones where the DNA is folded up, and then the combination of these techniques allows you to map between these nucleosomes. MNA-seq specifically focuses on mapping between the nucleosomes to understand where they're positioned. And I guess limitations at the moment for each of these techniques is that they're fairly low input, and not all of the map is delineated as yet. Yeah.
So I'm thinking about what James said. That suppose you have maybe this is too simple, but if you have a DNA that's kind of got a loop there,
Yep.
you sometimes get a haplotype that crosses over across the join and then skips the circle. So is there, especially, I'm trying to think of why there'd be extra linkage disequilibrium across the join. Does that piece sometimes across the join stay as a single piece? Or is there more linkage disequilibrium between things far apart when they're actually kind of near when it's three-dimensional?
I'm not sure the correlation between haplotypes and chromatin structure. I would say that it should be randomly located.
I don't think that is the case. I mean, I could imagine situations where you would need to have two particular alleles in two far apart places because they interact in some way that's essential for the survival of the fetus, and then you would end up with a high correlation potentially. But I guess that doesn't really make a lot of sense because if it just varies at all, you won't have any variation in those alleles. So.
Well, it could be some kind of epistasis across the things that are close when it's curled up in that way.
Yeah, I think that would be the only way that you would end up having linkage disequilibrium that jumps across the loop.
And during meiosis, so when this recombination is actually happening, DNA is actually very condensed. So it's not necessarily like these regions of open chromatin occur when the DNA is transcribing. Yeah, so. Okay, so we'll talk about DNA's hypersensitive sites and transcription factor binding. So as James mentioned, DNA's hypersensitive sites, basically they're sensitive to a DNase, which is a enzyme that can ligate. And transcription factors are proteins which bind to DNA, initiate gene transcription, and are really essential for regulating this gene expression. We also talked about iPSC, so the pluripotent stem cells, and obviously these are can go down a specific cell lineage by just adding a specific transcription factor. Multiple transcription factors bind to DNA temporally and also spatially control DNA expression. So this is a picture here of the DNA when it's condensed. When the chromatome is decondensed, you can have a distal transcription factor acting together with a number of different factors to regulate this transcription. Measuring transcription factors, mapping actually the sites that they bind at is really a vital step to construct a network of transcription regulation and, for example, help us understand the local SNP effects. The main method to do this is ChIP-ChIP and ChIP-seq. The limitation at the moment is that a lot of transcription factors don't have corresponding antibodies. A lot of the work has gone into mapping transcription factor binding sites. This correlates very well with DNA's hypersensitivity sites, so that's been useful. The latest estimate, how many transcription factors do we have? About 500 at the latest count, and this is based on 133 different human cell and tissue types. And importantly, each transcription factor had over 4,000 transcription factor binding sites associated with it. These transcription factor binding sites are also highly clustered in distinct human cell types, and you can classify them as being low, medium, high complexity. So you could have, this is just a picture of something that's in low complexity and something that's much higher. And so if something's higher complexity, there's more factors that are regulating a particular transcription of a gene. So from here we'll talk about methylation. So what is epigenetics? How is it regulated? So epigenetic regulation refers to a change in the genome that does not involve a nucleotide or sequence change. So you're not changing the DNA bases, but you might be either adding a histone or methylating the outer part of the DNA. And this can change the way the DNA is structured. So euchromatin refers to a loosely packed DNA, heterochromatin refers to a tight pack, and these are marked by different histone modifications. So a histone is so this is a picture of a nucleosome. So a nucleosome is actually 8 histone proteins that are all situated together, and this is what the DNA wraps around. It's protected in this sense. These histone proteins can be modified, so they can be acetylated, methylated, or phosphorylated by families of enzymes, and this can change the DNA structure. An example of this is the mark called H3K4. So when this is trimethylated and overlaps with promoter regions, it typically contributes to the recruitment of transcriptional machinery, and this tends to be active. Alternatively, the H3K47, when this is trimethylated, it's tightly associated with inactive gene promoters and really acts in opposition to the methylation of the H3K4.
Was methylation all on the histones?
No, so it's DNA methylation as well. So this is a picture of the histone being modified by methylation, and then we'll just talk about DNA methylation next. Yep.
Should telomere erosion be considered an epigenetic regulatory change or just a structural change?
So can you repeat that again? The
Telomere erosion?
telomere erosion, is that?
Should that be considered an epigenetic regulation change or just a structural
I'm not sure how telomeres are eroded.
Over
They just get
mitosis,
shorter
they
over-
lose, they just get shorter over time.
I'm not sure if that's related to epigenetic changes. Yeah, I'm not sure. But yes, it changes with age. Methylation changes with age. So, okay, so a lot of the epigenetic research in genetic studies focuses on this DNA methylation. So this is the addition of a methyl group to the DNA here. So this happens mostly at CPG sites. So I'm not sure, does anyone know what a CPG site is? So basically it's just when you have a dinucleotide of C's and G's. So the P just stands for a phosphate. So this is a cytosine and a guanine. So the modification, as I've mentioned, is outside the helix, but it's recognized by binding proteins to actually alter the chromatin structure and gene expression. So 70% of CPG sites are methylated, so they're a good indicator of regions of the DNA that will be changing in response to methylation. And these CPG sites are found in the promoter regions of many genes, but these tend to be protected from methylation. So this is a picture of CPG sites in the genome. This is a picture of what we would refer to as a CPG island. So a CPG island, there's different definitions, but basically there's a high proportion of these CPG sites within a certain length of the DNA. So this might be about 200 base pairs, and maybe 50% of those consist of these CPG sites. And so these these CPG islands are found in the promoter regions of many genes, and they're protected from methylation because obviously they're regions that need to be active for transcription. DNA methylation changes over time, and there's
just a question about that.
yes.
So they're protected from, so they already are methylated in these CPG islands? They have a lot of methylation already?
70% of CPG sites, when they look like this, are methylated, but CPG islands are protected from methylation.
But in that picture, what does it mean that they're highlighted? Does that mean that there's a
That's the
methyl group on-
CPG site, and it means so in the picture, all of the CGs are highlighted in yellow. So this particular square here doesn't have a very high proportion of CG sites. There's much fewer in this length of the DNA compared to this length. So this would be referred to as a CPG island, and it would be protected. You would expect it to be protected from methylation, and then, but not in this case.
Why is it protected from methylation? I mean, how is it?
Typically, this occurs in the transcriptional start sites.
But how is it protected? Like, why is it that the methyl groups can't attach there?
I'm not sure.
Okay.
I mean, they do form a special structure, I think.
Okay.
Like just like the way the DNA binds to itself. So like, I'm not entirely sure, but I think that structure maybe protects them from getting methylated. I'm not sure.
Yeah. Typically, I mean, they're open. Yeah, I don't know. But yeah, methylation is it changes over time. There's and this has been mapped out through embryo development. I think this is data relating to the mouse. So fertilized embryo goes through these waves of large changes in DNA methylation as the embryo is developing. And a lot of this epigenome variation is controlled by genetic mechanisms. Yep.
So once a CPG site is methylated, can it be unmethylated?
Yes.
as
Yep. Okay, so the next few slides is just talking about methylation arrays. So these are kind of, I guess, in a way similar to SNP arrays. So they're pretty commonly used. basically it's a chip that you can measure CpG DNA methylation. Over time, these chips are similar to the SNP arrays. There's more CpG sites that are actually getting measured. The most common in the literature, I guess, is the 480K chip. And then this is being discontinued. I think they're trying to pick up an EPIC chip. So this is double the number of sites. And this is a common high throughput approach to kind of measure changes in methylation. Yep.
Are there data sets that include both data from methylation chips and from SNP chips, or is it usually separate?
Yep. Most data sets would have both because it's a fairly cheap method, both SNP arrays and methylation. And because we know that there's a genetic influence of the methylation, I think most people are starting to think that both need to be considered together. Yep.
For the data sets that most of us would use, there's very few of them have methylation data so far, and the total available sample size with methylation data is much smaller than with GWAS data. But ALS-PAC is one important data set that has both. The health and retirement study does not yet have methylation data, but I believe that they're working on it. So, you know, it's going to vary by
Cohort.
data set.
What's the first one you mentioned?
ALS-PAC.
ALS-PAC,
ALS-PAC.
the Avon longitudinal study of parents and children.
Wow. Which were the 90s.
Okay, stand corrected. You should talk to Alex about that one. He's the resident expert.
Yep.
Isn't it also the case that methylation, I mean, it varies based on the tissue type more so?
Yes.
And so, I mean, most data sets are just collecting saliva, and trying to do epigenetics off of saliva is not ideal. I mean, it's better to do it in blood. So that's an additional hurdle for a lot of these data
Yes.
sets to
Yeah.
collect, I mean, a lot of these like population
Yeah.
representative.
Yeah, I think you make a good point that methylation is similar to DNA sequencing, but there's a lot of variability in it, which can alter your result. And so I'll try to highlight that.
Can I add something? to
Yes.
that? So I believe that methylation marks in saliva is closer to methylation marks you would expect in neural tissues than methylation marks in blood, because both saliva and neural tissues are from the ectodermal layer, or the cells that are there in the saliva that you use to sequence, look for methylation marks.
And even in blood, we need to correct for cell types. There's many different cell types in the blood, and that's part of the QC of the methylation data. To measure methylation, you want DNA that's high quality, and that's best often from blood or tissues. You want batch controls and technical replicates. You are at a higher risk of bias and quality control issues than DNA sequencing. The methods, you can use region-based or single-base technologies. The gold standard is bisulfite sequencing. So what is that? Basically, when you expose a single-stranded DNA to bisulfite, if the region is actually methylated here, it will remain as a cytosine. If it's unmethylated, the cytosines will actually convert to a uracil. And then you pretty much just compare pre-post this treatment. Bisulfite treatment is really damaging to the DNA. So that's a problem why you need this high quality DNA input. So once you treat the DNA, then you'll sequence it either using next generation or SNP arrays, and the arrays make use of these fluorescent probes, so like a hybridization approach. Experimental design is critical for reducing and controlling for technical variation. Yeah.
So methylation can vary across different tissues and can vary across different cell types.
Yep.
Within a cell type, is methylation of DNA relatively constant?
I'd say yes, relative to other cell types. I mean, there's always internal variation, but that variation is less than between cell types, I think. Yeah. There's a lot of large databases that are now trying to profile a lot of tissues, so like tease out the heterogeneity between tissues, maybe also within tissues. So it's a constantly moving field, I think.
And the correlation across tissues, which is particularly important for us if what we really are interested in is methylation in brain tissue, but what we have is methylation measured in saliva tissue or blood tissue. You know, we want to know how reliable
whether it's a good proxy or not. Yeah. And that's the same in expression studies.
What do you know about these correlations between methylation in different tissues? If we want to get methylation in brain tissue but can't, is it right that saliva is closer than blood, and is there anything closer than saliva that you could imagine?
I'm not sure personally, but I'll bring up some resources that have profiled these in these different tissues. So
I think it's actually a slightly controversial area of research. It's very active right now, and you know, I've seen papers arguing kind of both directions. I don't think it's quite settled yet how good of a proxy it is.
Yeah. I'll just talk a little bit about experimental design. This can be applied to SNP arrays, of course, but it's particularly important if you're looking at methylation. Cases and controls should be collected using common methods, so over a similar time frame, have a similar demography, things like age, sex, ancestry, smoking, because these are all things that affect methylation status. The DNA should be extracted and actually stored by a common method, ideally not in batches and cases and controls, so, you know, together as one. Ideally, you want to record all the information on the DNA extraction. You want to randomly place cases and controls for the bisulfite conversion, randomly place them on arrays, and also consider using control samples and duplicates to track quality over time. So these are important points that, because our lab has worked quite a lot with this methylation data, this is things that they've picked up along the way that are important and are helpful. I guess, you know, if you're thinking about designing an experiment, for example, say you want to investigate the transmission of DNA methylation across generations. So you've got mothers and daughters. So how would you actually place them on your chip array? What would you not do? Well, you wouldn't put the mothers next to the daughters. You wouldn't put all the mothers on one side and all the daughters on the other side. You wouldn't put mothers on one chip and daughters on the other chip. So you should randomly place your cases, controls of interest. So.
Why don't you want them next to each other?
Because the way methylation, it's a fluorescent reading, and there's even bias in rows when you read these chips.
Because like neighbors can contaminate the readings of each other, is that?
No, for example, row 1 typically is a different brightness to the bottom row kind of thing. So there's an association with placement on the array. So it should always be randomly placed. You shouldn't always be placing cases on the left side and controls on the right side.
Okay.
I know it sounds unusual, but this is what our lab has learnt, and this is different from SNP arrays, which are a lot more controlled, I think, and less variable in this methylation process. Yep.
So you mentioned smoking affects methylation. Are there other like social science phenotypes that actually affect methylation as well? So like behaviors that people do that can actually
Aging?
well,
Yeah,
so
aging and smoking are the most well replicated across cohorts, and it's very consistent in that finding. I'm not sure about other phenotypes and how consistent they've been.
So I have a paper with a number of co-authors where we're just looking at methylation association with educational attainment. And what we're finding is that there are a few associations that come up in a sample of about 10,000 people, because by the way, these methylation associations tend to be much, much larger in magnitude than SNP associations. So they can be detected in smaller samples. But the magnitudes that we're getting are about an order of magnitude smaller for education than the associations for smoking and other kind of more direct biological outcomes. And also, they all the ones that we can detect seem to be confounded by smoking. So they're basically all in methylation sites that are also known to be related to smoking. And even though we have, controls for smoking, it could be that the most likely explanation for what we're seeing is that people who are more highly educated are less likely to have mothers who smoked, and mothers smoking affects methylation of a fetus. So there's high potential for confounding. That's just one study and one phenotype. But my reading of that evidence is that for kind of the complex behavioral phenotypes that we're often interested in, we should be very cautious about making too much of methylation associations, and it's looking, at least like the initial evidence is that those associations are much smaller than things that have direct biological impact.
And I think the most paradigmatic example, Remy, that I've seen is Rachel Yehuda's studies on Holocaust survivors and methylation, although some people question some results. But I think she's documented, she's published in American Journal of Psychiatry on how Holocaust survivors have intergenerational transmission of epigenetics.
Yeah, so that's stress. High stress is definitely related to methylation, and that's an extreme version of stress.
Yeah.
Okay, so this is just an overview of what these SNP arrays actually cover. So, for example, on the 450K array, a lot of the sites are, they cover all of the CPG islands. So these are the regions typically around these transcriptional start sites. Also, they refer to things as shores and shelves. The EPIC is coming into use now, so this has greater coverage of these regions. But in general, the 450K is actually pretty good at covering regions of interest, basically from a methylation point of view. This is just two examples of methylation and age. So DNA generally becomes more methylated as we age. This is just some twin data from quite a while ago now, but you can see 3-year-old twins, methylation is quite low, and then 50 years later, the methylation is increased. Horvath in the genome biology paper in 2013 was actually able to identify about 350 SNP CPG sites, which very accurately predicted age in independent samples and multiple tissues. And so this is just data from our lab. We've got some cases and controls. We've just plotted the DNA methylation age versus their chronological age, and it's slightly below this linear line here, but the correlation is actually pretty good and can predict within plus or minus 10 years. Yep.
So this evidence suggests that one way you might be able to deal with things like age and smoking is think of it a little bit analogous to these principal components. If you just took out the principal components of methylation, you'd get age, smoking, a few things that you hadn't realized were a problem, and then kind of trust the rest. It's not that it's the same issue as population stratification, but the same solution might work.
So were you just saying that you tried to control for smoking methylation, but it still didn't?
No,
you weren't able to.
Smoking methylation, we controlled for smoking, like actual self-reported
smoking,
which is noisy, so that's one problem that, you know, we may not have, controlling for a noisy measure of smoking doesn't actually eliminate the effect of smoking. And the other problem is that parents, you know, people who are less educated are more likely to have mothers who smoked, which affects one's own methylation. But I think Miles is suggesting using the methylation data itself to create some index that might actually be a very good marker of smoking. And I don't think that's been done, but I think it's a cool idea.
All right, so with this methylation data, you can carry out things called epigenome-wide association studies. The only thing to note here is that unlike DNA, methylation can change over time. And so basically, at a cellular level, a change in methylation, yes, can cause a disease, but at the same time could be a result of the disease. And probably the best study design would really be longitudinal, where you have a population of people, you collect longitudinal data on them, and then you have a proportion that become cases and controls. But obviously this is quite difficult to do, and the more typical study designs is within families and cases and controls. Yep.
So how should I think about the units of methylation? So it's just some percentage of the DNA that's methylated, or does it matter more that certain areas are methylated than others?
I probably, the most simplistic way to think about it is just similar to a Manhattan plot. So you're getting if you're just doing a case control study, then you just look for methylation, whether that be up or down in cases versus controls.
But I think the question is about the measure of methylation. So when you measure my methylation at a CPG site, we get a continuous number. I think the question is, what is that number?
So it's a fluorescence measure, so whether a site is methylated or unmethylated.
Okay, and then those are just aggregated over the whole
Yep.
genome pieces that I'm looking at, just additively, so I just add up the fluorescence at each site to get kind of total amount of methylation?
I'm not sure. Look,
you get a methylation level at each site.
Yeah, so I'd say you're either methylated or unmethylated, and that fluorescent value would allow you to distinguish between the two.
The typical methylation data gives you basically a vector, long vector, 450,000 or whatever the number is in the array. So you have this, it's like the set of SNPs, but this is the set of CPG sites in the genome. And that each of those you have a continuous number, which is some measure of methylation. So the reality may be that it's either methylated or not, but the measure that you get of that is a continuous number.
Should we think of it as like probability of like what does it mean for something to be more fluorescent? Like how should I interpret that if something fluoresces a whole lot?
Well, it's not whether it fluoresces or not. It's a ratio between green and red fluorescence. So methylation is measured using, in the latest Illumina array, using these, what they call type 2 probes. So these are pretty much a bead, and it's got a probe for a methylated site and unmethylated site. And so the methylated site, I think, is red, and the unmethylated site is green. So if the site is more often found, then you'll have a green fluorescence, and then if it's more unmethylated,
but then it's still a continuous number, right? So is that number just how red it is, or and?
Well, I wouldn't say it's a continuous. I'd say it's either
Let
red.
me look up the answer. Why don't you go on?
Okay.
Look up what the answer is.
Sorry, can I ask you one question? So
Yeah.
on the graph that you showed, that you plotted age against methylation.
Yeah.
Methylation is plotted on one axis.
Yes.
So that means that there's been some aggregation of the methylation measure across the different sites in the DNA.
Yes, we've taken the measure of 350 CPG sites.
And do you just average those, or do you just add them? Or?
I haven't done it, so I don't know, but
How do you
it's
create
based on the
that?
methylation status of these 350. So maybe some are up, some are down, but
It's what you mean, how he creates the score.
Yeah, exactly.
I mean,
was
it's
wondering-
it's like a polygenic score. So he's trained these sites, and they're like the betas from a GWAS, but they have certain predictability with with age. And then he puts those in a sample that wasn't used to predict the betas to sum up, kind of, predict age in that sample, right? I mean, similar to, that's my understanding of it.
So the answer to the question of what you get on these chips is the proportion of methylated molecules at the CPG site. So it's a number between 0 and 1.
Because there's like heterogeneity across the cells, and so there might be some that are and some that aren't. Is that?
So I don't know, I'm not sure why you have more than one molecule at the site.
So-
Methylated.
I want to ask, so the other day, when you gave your talk, you were saying a great advantage of genes is that they're determined at conception long before all of these other social outcomes. And of
Yeah.
course, that's not true for the methylation. So what would be your thinking about why is methylation data cool? It certainly doesn't have exactly that characteristic of the nucleotide genes.
Well, I think, so methylation right now is hot. I think it's, both in terms of funding agencies' interest and in terms of the popular press. And I think the reasons why it's hot are mainly that there was this very interesting discovery that at least some methylation, the methylation of the sperm or egg cells can be transmitted from parent to offspring. That's potentially a mechanism by which environmental effects can carry through to future generations. If environmental effects cause methylation and then the methylation gets transferred from parent to child, that's a kind of Lamarckian mode of inheritance that we thought wasn't possible, but now we know it can happen through methylation. So that got super exciting. And the other thing is that it's kind of it's politically correct, because it's a way of the environment. It's instead of saying the genes are these fixed things, people have this misconception that the effects of genes are fixed and deterministic, and now we have this thing we can study where we can see that the environment changes the way that the genes operate, so it's not actually fixed after all. I think that there are also, interesting scientific reasons to be interested in methylation, because we learn a lot about the actual process by which gene regulation happens, and a lot of, some of what we think of as gene-environment interaction may operate through methylation channels, so the environment may affect the expression of genes that I think Fleur's going to talk about.
Yep.
I think that the excitement about the methylation getting passed on to children is overhyped, because it's actually just a tiny fraction of methylation where that happens. It's only in the sperm and egg cells, and even then, most of the methylation is wiped clean when the embryo is formed. It's only in some places where it can remain, and I think the evidence in humans is that there isn't really evidence yet in humans of any kind of transmission, as far as I'm aware. And I also think this is, as you said, Miles, because it's not fixed and it's affected by the environment, it's much harder to study methylation than DNA. You don't know which direction, you have all the same kind of issues of correlation versus causation that come up when you don't have predetermined variables that aren't randomly assigned. So I think our state of knowledge about methylation is much more primitive than our state of knowledge about DNA
Yep.
and the effects of SNPs at this point.
Yep. We don't have a lot of time, so I can briefly mention this. This is just a recent study that has done an EWAS and also combined this with what they've termed a TWAS, so transcription-wide association study. This is in smoking. This is the Manhattan plot. This is their Bonferroni corrected. They've listed the top 10 genes and those that are common between the altered methylation and the expression. There's other ways to look at methylation data. You can do gene set analyses, and obviously you need to correct for CpG sites per gene, because obviously these differ. And then another way to analyse methylation data is actually look at differentially methylated regions. There's a few different methods to do this. You can use a window approach and look for kind of bumps. So this is combining SNPs and their methylation status to combine the evidence of association across multiple methylation sites. This can improve the power if a region has multiple independent signals. And this window approach, I guess, probably has been more commonly used in the cancer fields. These are the two databases that are profiling different tissues and looking at methylation status. And so there's actually, and this is a ever-changing source of information. I won't talk about genomic imprinting and X chromosome inactivation in terms of time, because we've got to talk about RNA seq and expression, and that's probably more relevant. But that information is there, I think. So the PDF of these slides are actually in the Dropbox now, so you've got access to those. A quick summary, and before we move on, there's multiple different controls involved in the regulation of gene expression. ENCODE is a very good source that aims to annotate these regulatory regions. DNA does not function linearly and can form connections across the length of a chromosome. We've talked about methylation, so this can occur in the histone modification and also on the DNA. And this can actually alter the structure of the DNA to activate and silence gene transcription. You can measure this genome-wide to examine the cause and effect of a trait or disease. Genomic imprinting and X activation are just two methods that occur in the body that result in monoallelic gene expression. So for the rest of the talk, I'll talk about, briefly go over gene expression, protein expression, and I've just got one slide on metabolomics. So what is the interest in gene expression? So this can, as James mentioned, help you prioritize causal gene follow-up, in which genes that harbour mutations can affect expression, which in turn affects phenotype. So this is a diagram of a genotype influencing transcription, influencing a phenotype. So a recent example is on the case of how likely it is that the causal locus or loci is actually the closest gene. So Jian Yang was the last author in this particular paper. So he looked at 5 different complex traits using GWAS data. This involved up to 300,000 individuals, and he also used QTL data on 5,000 individuals. So this is all expression data from blood. From this, they were able to prioritize 126 genes. And actually, of those 126 genes, 77 of those were not the nearest annotated gene to the top GWAS SNP hit. So I guess this is an accumulation of evidence that is starting to show us that the DNA is not acting linearly. So if we have a SNP, it's not necessarily influencing the expression of the closest gene, but it might actually be further away than that. The other way you can use expression data is to do a pathway or gene set analysis for looking at disease mechanisms. And so there's different methods to do this. Some are described in this picture here, and it really depends on your question of interest. So how do we measure expression? So messenger RNA can be measured very accurately and consistently in tissues and cell lines in humans. I think microarrays have been used routinely. So this is a hybridization approach. You extract the RNA, you convert it to cDNA, and this is used as the input. And then this hybridizes with sequences on the chip. So this is a highly reproducible method, and then typically if you're looking at differential expression, it's often shown as a volcano plot and demonstrating changes, so genes that are up versus genes that are down when you're comparing two groups. Microarray though is not no longer being used. This has kind of been superseded by RNA expression. I've just got a picture here of the heritability of different types of tissue and their expression. So if you think of traits as 50% of the genetic variance for gene expression is not being tagged by common SNPs. Of the variance that is being tagged by common SNPs, quite a large proportion of this can be attributed to identifiable QTLs. Okay, so RNA-seq. So here's a picture of the overlap between microarray and RNA-seq. The overlap is actually pretty good. However, it's pretty obvious that RNA-seq is picking up a lot more transcripts compared to microarray. So the dynamic range and sensitivity of an RNA-seq approach is much larger, and you can detect a lot of novel transcripts. Using RNA-seq, you can measure genes, exons and transcripts. And there's a lot of different tools to help you analyse this type of RNA-seq data. I won't go through the methods. I'll briefly talk about DEPICT because James has already spoken about its use. It is a fantastic and integrative tool that's been regularly used to predict gene functions and systematically prioritise the most causal genes at associated loci, and also highlight enriched pathways that identify tissues or cell types where genes from associated loci are highly expressed. So there's a lot of available resources to look at expression. This includes GTEx. This is actually a really friendly web interface where you can interrogate a particular loci and look at its gene expression across different tissues and its relationship with genetic variation. So individuals have been collected and with multiple tissues types, and they've also been genotyped as well, and so you can look at the association with expression and across different tissues. CAGE is also a database that has the limitation, I guess, with GTEx is it's limited in the numbers of people. I think the blood expression databases are much larger, so you're looking at more than 2,000 to 5,000 instead of 500, which I think is in GTEx. There's also some brain expression databases. There's a move towards to do single cell sequencing, and also summary data-based Mendelian randomization is also a good software tool to kind of test for an association between gene expression and a complex trait. So the last two sections I'll talk about is proteins and metabolomics. So proteomics is the study of the proteome, which is describes the full set of proteins expressed by an organism. And so when I was talking about microarray and RNA-seq, these are actually measuring transcript levels. And so these are a lot easier to measure than protein levels. But changes in protein could have more or greater functional significance. Proteins exist in a 3D folded conformation, which is required for their biological activity. I guess the questions in the proteomic research is what is the finite list of proteins, where are they expressed, what is their rates of production and degradation, their steady state abundance, how are they actually modified? So there is post-transcriptional modifications that occur, such as phosphorylation, sulfation. What is the movement of these proteins across different subcellular compartments? What is their involvement in the metabolic pathways and how proteins interact with each other. Capturing proteins in tissues can be difficult. Bone is a good example. It's very resistant to enzyme digestion. Typically antibodies are used to measure proteins, and obviously there's a lot of limitations with antibodies, and this has been debated in the literature. I think there's a move now towards a more high throughput approach, which is mass spec, and I won't go through the methods, but it is improving detection, but there is still, it's still not as high throughput as something like RNA-seq, which is really, I think, the best way at the moment to characterize expression. This data has been merged, so they're looking at both transcript and protein levels in a whole lot of different tissues, and this is a nice paper that does both, so it's a huge amount of work. They've looked at 16,000 protein coding genes together with RNA-seq data for 32 different tissues, and they've included a genome-wide analysis of tissue specificity of the RNA and the protein expression. And interesting, like they found that there's actually quite a high proportion of ubiquitously expressed genes. So there's obviously a set of genes that are expressed in a lot of different tissues to maintain important roles in cell structure or translation, transcription, and replication. When you're thinking about protein expression, it's good to recall that eukaryotic cells contain numerous compartments, each which have different protein constituents, and proteins targeting the same subcellular localization tend to participate in mutual protein-protein interactions and are often functionally associated. So you can use this information about where the protein is actually localized to see if there is an enrichment in disease or complex traits. And I think James mentioned this this is part of what DEPICT can do for you. So this is just an example of different protein classes, and this is an example of where they might cluster. I put this in because this is something our lab is actually starting to think about, and that's the translation of genetic information. So in drug development, they're interested in improving or increasing the probability that a therapeutic agent engages in a target protein and at safe and efficacious doses in the relevant tissues to prevent or treat disease. And over a quarter of drugs that actually enter clinical development fail because they're ineffective. And a recent paper came out to say that 61% of approved drug indications had at least one genetic association with a related trait. So pretty much what this was saying is that drugs fared better in the pipeline when they had this, when they were supported by genetic evidence. So I guess when GWAS was started, they were like, well, what is the point of this GWAS? How is it useful in a translational aspect? And so there's kind of a renewed interest, particularly in the drug development area. And most pharmaceutical drugs actually act by targeting proteins and modulating their activity. So these are things like receptors and enzymes. So if you have a trait or disease that is associated with a protein, then of course it could be a good candidate for translation. So obviously there are other lines of therapy that are being investigated, you know, more epigenetics and RNA, but at the moment most of the approved drugs are targeting proteins. So lipidomics and glycomics refers to lipids and carbohydrates. It is an aspect of metabolism. This is really an emerging field. So not a lot has been done for a number of reasons. I think the reason is that there is a lot of metabolites that are produced, and this is constantly changing between individuals over time, and there's no good way to actually measure every single metabolite. So functionally they're of interest because coordinated anabolism and catabolism is a key molecular integrator of energy homeostasis, membrane structure and dynamics and signaling. And imbalances in metabolism can contribute to diverse phenotypes and disease states. So obviously metabolism is required for basic cell function, and so metabolic disorders often have system-wide effects. In terms of the brain, it actually has a disproportionate reliance on metabolism. So it comprises a smaller percentage, or quite a small percentage of total body weight, but uses quite a high proportion of the body's oxygen at rest. Yep.
So would something like Depict currently have a lot of annotations for things like glycomics and lipidomics and metabolomics?
Probably say no.
Oh.
But James, maybe, you know. Is there much metabolite data in Depict?
I mean, to show a slide, but I mean, one of the traits to which Depict has been applied is cholesterol effect, and the top results all have something to do with cholesterol synthesis, cholesterol transport. But I don't know what information is used by the curators to come up with these gene sets. It's this kind of thing up here.
Yeah.
So I don't know if there's that connection.
Yeah, there's.
Yes, but the cholesterol GWAS you put into the Depict, but it spits out as all the cholesterol, iron processes.
So yeah, overall, I think this area is very difficult to measure. It's not possible to comprehensively profile a cell or a tissue and measure every single metabolite. Just doesn't exist at the moment. And of course it changes quite rapidly. So I've just got some final examples of how these types of expression data have been brought together. It's an emerging field. So this is from Michael Inui, and he had data from Finland. He had fasting whole blood, and then they had matched genome, transcriptome, metabolome information on these individuals, and even though it was only about 600, they actually found some quite interesting data based on this this data set. He demonstrates a way to, so this is very high dimensional data, and he's has put it together in a, reduced these into kind of metabolite networks, which is a way to deal with this high dimensionality. He's also done things that involve doing like a weighted gene expression network. So where he's labelled, so he's collected different tissues from a mouse model, looked at the networks within these tissues, used nodes he's used as the genes and the probes, and modules as the subnet or clusters, i.e. known biological pathways, to kind of distill this down into usable information. I guess for all of you to know, integrative analysis is a relatively undeveloped area. There's a lot of scope for development and novel ideas, and there's no real consensus on analytical approaches and strategies to put all this data together. So I think it's probably, yeah, it's good for you to think about it coming from your background. His advice was it's important to understand the data, know the technology, be aware of the sampling, and of course explore. Because there are some difficulties in putting all this data together. So when we talk about genome, epigenome, transcriptome, proteome, metabolome, you know, it does become extremely complex. So, and there's a lot of resources that you can refer to. So, so just the final slide. I talked about sequencing technologies. So obviously these permit genome, epigenome, transcriptome, proteome and metabolite profiling. Experimental data together with the computational biology can really help to identify molecular mechanisms, underlying components of a complex biological system. This is starting to provide us with how a cell works and functions as an organism and how genetic variation is associated with traits and diseases. And obviously this permits quantitative description for function and hopefully, you know, findings to be translated in a precise way. So that's it.